So let's now continue with the thyroid hormone. The first thing is, maybe let's understand. The thyroid hormone is produced by the thyroid gland. What is the function of this hormone? It's a very important hormone, thyroid. It is involved in growth and development of the embryo, especially the development of the brain, okay, together with growth hormone. It also helps to regulate internal homeostasis in the young. For example, it will increase the basal metabolic rate. Thyroid hormone is involved in, regulation of internal homeostasis by increasing the basal metabolic rate. This is the rate at which you are able to work, the energy that you are spending, the body temperature. It also helps maintain metabolic energy balance by increasing the number and size of mitochondria, increasing the enzymes in the electron transport chain, also increasing the sodium potassium ATPase activity. Generally, the thyroid hormone is excitatory for normal cellular function, including heart muscle. It basically excites the body, and the body will spend more energy if the thyroid hormone is released. As we said, it is released by the thyroid gland. The thyroid gland is one of the largest pure endocrine gland. It is about 20 grams in weight. Its size will depend depending on the age. It will increase with age. As you increase in age, the thyroid gland also increases. For the females have got a larger thyroid gland than the males. And then the size will also depend on physiological conditions like the pregnant women and those who are breastfeeding. It will tend to be much bigger because they are spending more of the energy. Where is the thyroid gland located? It's located in the neck, just below the larynx, on both sides of the uh, and anterior to the trachea. So this is where the guy is. You can see this where it is there. Thyroid gland is well vascularized. It is rich in blood supply. So what is the blood supply to this gland? You can see this is the arc of the aorta. The first branch from the arc of the aorta is the brachiocephalic trunk, to divide into the right subclavian and the left subclavian. The second branch of the aortic arc is the left common carotid, and then you have got the left subclavian. So the common carotid arteries, they are going to bifurcate. Can you see that it bifurcates into external carotid and internal carotid? So the external carotid is going to give what we call the superior thyroid artery. And then the subclavian artery, this guy, is going to give what we call the thyrocervical trunk. This trunk here. Thyrocervical trunk is going to give the inferior thyroid artery, which will supply the inferior aspect. And then the brachiocephalic trunk itself can as well give what is known as the thyroid inner artery. This artery that you can see here, that is supplying the middle aspect. When it comes to embryology, the thyroid gland comes from an invagination of the floor of the pharynx when development is happening, the floor of the pharynx. And the thyroid gland has got two lobes, the right and left. They are connected on the middle, which is known as the isthmus, on the middle there. So the normal thyroid is known as the euthyroid. Can you see that it has got multiple follicles? This is a thyroid follicle, thyroid follicle, thyroid follicle. There are a lot of them. They're also known as acini. These are the functional unity of the thyroid gland. They are about 100 to 300 micrometers in diameter. And each follicle is spherical in structure and is lined by cuboidal epithelium that secrete into the interior of the follicle. This for cuboid epithelium is what will be producing the products into the interior. And each follicle is filled with a pink staining proteinaceous material which is known as a colloid. So inside the follicle, I've got a colloid. Can you see the colloid there? Don't forget the epithelium, don't forget what is inside. So when the thyroid gland is inactive, the colloid is going to be very much abundant. The follicles are also large. The lining cells are flat. The cells which are lining are flat. But when the gland becomes active, the follicles are going to become small. The lining is going to become cuboid or columnar, and then the edge of the colloid is scalloped. It will form many reabsorption lacunae. Can you see that just opposite to the thyroid, to the epithelium of the follicle, you have got the C cells which are producing calcitonin. And each follicle is surrounded by good and rich blood supply. The thyroid cells are found on, they rest on the basement membrane, the basal lamina. The prominent endoplasmic reticulum is a common feature in most granular cells, of course, endoplasmic reticulum, and secretory droplets or thyroglobulin is going to be present. We are going to understand what we are calling thyroglobulin. When you are forming the thyroid hormone, it is a nice mechanism. Remember last year's exam, that's why they just brought right about all the steps of thyroid hormone synthesis and draw a diagram, 20 marks. Okay. So the colloid, as we said, is a gel-like substance that contains glycoprotein, which is called the thyroglobulin. Remember the colloid is this pink part. It contains thyroglobulin. Thyroglobulin stores the thyroid hormone within its molecules. You also have parathyroid hormones, which are just next. These are spherical cells which has no relation to choroid or cuboidal cells. These ones, they secrete calcitonin, which is involved in the metabolism or homeostasis of calcium. Now, how is thyroid hormone secretion happening? So there are two types of thyroid hormones. You have got T4, which we call thyroxine or tetraiodothyroxine. Thyronine, sorry. Tetra because of the four iodines, tetraiodothyronine. You also have T3, which is also known as triiodothyronine. Please take note of that. So these two hormones are what makes up what we call the thyroid hormone. Thyroid hormone is secreted by the follicular cells, the cuboidal guys, and can be stored in the thyroid gland for a couple of months, even up to 2 to 3 months, having significant effect on increasing metabolic rate of the body. Calcitonin is secreted by the parafollicular cells or the C cells, these guys, okay, and it's important in calcium metabolism. What is the chemical nature of the thyroid hormone? T3 has got two benzene rings. You can see these benzene rings like that. And then you have got three iodine atoms. That's why you call it triiodo. So one of these benzene rings has got only one iodine, and then two, the other one has got two. And then T4 has got two benzene rings with four iodines. You can see that's why it's known as T4. So when thyroid hormone is being secreted, majority of the hormone is secreted as T4 thyroxine, 93%. Only 7% is secreted as T3. But then what happens is that T4 is not active. Almost the entire of it is converted to T3 because T3 is the active form. So T3 is 4 times potent than T4. In other words, it is 4 times more active than T4. But it is present in smaller quantities in blood and persists for a much shorter time than does T4. So T4 is found more in blood. T3 has got a greater affinity to nuclear receptors than T4. So when you have got even a little T3, you'll be able to attach to the receptors in the nucleus. Remember we said that the thyroid gland is producing the thyroid hormone is also part of those hormones which are hydrophobic. So meaning the receptors are inside the nucleus. Okay. So almost all the thyroid hormones are going to be carried in the blood in the inactive form. And they are bound to three different types of proteins. Please take note. Thyroid hormone can be transported binding to globulin. The majority of it binds to globulin, 80%. Some of it, 10% to bind to prealbumin, and then some of it will bind to plasma albumin. T4 has greater affinity to bind proteins than T3. Because of this, that is why T4 is actually inactive. Because when it is bound to these proteins, it is just there to be transported. It's not free to wake. That's why it's inactive. And only very little T3 and T4 are carried in the blood in the free form. Majority of the hormone is carried bound to any one of these three guys. Please take note of them. So how does a thyroid hormone work? It is a lipophilic amino acid derivative. So the receptor is found in the nucleus. It is an amino acid but it's lipophilic, right? So you can see when it is hormone is transported in blood bound to its carrier protein, it's going to pass the membrane and then it is going to be able to go and attach to the receptors in the nucleus and then exert its function. So look at this, what is happening. You have got in blood, you have got a carrier protein for T4, which is going to release the T4 and then it is going to diffuse into the cell. In the cell, it is going to bind to binding protein. That binding protein is going to transport it and then it is going to release it. It's actually the T4 is first going to be converted to T3, which is going to bind to the binding protein, and then it will now be released into the nucleus. The nucleus is going to stimulate the nuclear receptors, and that will lead to mRNA production, and mRNA can form proteins. That protein we are talking about is actually the thyroid hormone. Okay, that will be the response of the thyroid hormone. For example, increasing the basal metabolic rate. So we need to know how is the thyroid hormone synthesized. These are the steps. Please make sure that you know them. All right. Thyroid hormone is synthesized in the colloid by number one. There is going to be thyroglobulin formation, and then iodine pump or trap, and then oxidation of iodine to iodide to iodine, and then iodination, condensation or coupling, and then thyroid hormone secretion, and then deiodination. So these are again the steps, the seven steps which are here. So the first step, step one, is the formation of thyroglobulin. Thyroglobulin, as we said, it is found in the colloid. It is a glycoprotein which is made up of two subunits. It is synthesized in the rough endoplasmic reticulum and the Golgi apparatus of the thyroid cell, following the entry of amino acid tyrosine from extracellular fluid and then incorporated into secretory vesicles. It is going to be secreted into choroid by exocytosis of granule that also contains thyroid peroxidase. So you can see this is your thyroglobulin molecule, and you can see to the thyroglobulin molecule are going to have tyrosine binding there. Okay, so that is how you get to make this thyroglobulin. You have got tyrosine attached to it, two of them. Some of them they've got only one tyrosine. The next step is going to be iodide pump. So if you can even see here, step one was the formation of thyroglobulin. For you to form thyroglobulin, you can see you have got thyroglobulin here. You need tyrosine also to attach there. Okay. And then there'll be iodine trap. This is what you can see here. You can see this is iodide. It is an ion. So it is trapped from the blood. Can you see that in blood? You are going to use an iodide sodium transporter transporting sodium and iodide. When it is trapped, it's now going to be taken into the lumen of the follicle. It's going to be converted to iodine by the peroxidase enzyme. Iodine. From iodide to iodine. So we are the iodide pump. Okay. So iodine is found in food, salt, and seafoods in the form of iodide. 120 to 150 micrograms of iodide is needed daily to be able to maintain normal thyroid function. The iodide is going to be pumped together with the sodium, and that is known as trapping into the follicles. And that action is stimulated by the thyroid stimulating hormone, which comes from the anterior pituitary gland. Remember, the first the hypothalamus is going to stimulate the anterior pituitary gland by releasing thyroid releasing hormone. Thyroid releasing hormone is now going to cause the release of thyroid stimulating hormone. Thyroid stimulating hormone will cause the release of thyroid hormone. But then what is happening that the thyroid stimulating hormone is acting on the thyroid gland to cause the trapping of iodide. Iodide will be 30 times in thyroid cells greater than blood concentration. So in other words, you're going to have more of it in the thyroid cells than in blood. So there's what is known as a Wolff-Chaikoff effect, a reduction in thyroid hormone levels caused by administration of large amounts of iodine. Remember, when you have got a lot of iodine, it will send a negative feedback to the brain to say, okay, enough of what we are producing. So that's known as a Wolff-Chaikoff effect. When you have the iodine and you have got the thyroglobulin, then you are going to have oxidation of iodide. Remember, the first one was trapping of the iodide, just getting it into the cell. So now we're going to have to oxidize it. So it was iodide, you want to convert it to iodine, and what you use is the peroxidase enzyme. Okay. And then the next thing now will be iodination. It's also known as organification of the thyroglobulin. So what happens that just the iodine that you are formed now, you are going to add it to tyrosine. The tyrosine that is found within the thyroglobulin, remember, thyroglobulin has got tyrosine. Okay. These two lines that I've put here, those are tyrosines. We can add one iodine or two iodines to each tyrosine. So if we add two, that will be known as the DIT, diiodotyrosine, because I've got two iodines to one tyrosine, or monoiodotyrosine MIT. So these ones are going to remain attached to thyroglobulin until the gland is stimulated to secrete the hormone. What is going to happen the next step will be now the condensation or coupling. In coupling, what is just happening is that the MIT, remember this is monoiodotyrosine and the diiodotyrosine. What can happen is that you can get the mono and the di, you couple them, you add them together. So they are going to form now you are going to have three iodines, right? MIT, monoiodo, and diiodo. You are going to have triiodo. So that is what forms the T3 enzyme when you couple MIT with DIT. For you to form T4, which is thyroxine, you need to couple DIT with another DIT. Take note that not all DIT and MIT will form the thyroid hormone, only 25%. The rest will remain like that. T3 can also be made. So when you have formed T4 from coupling DIT and DIT, you can as well now convert By deiodination, we can remove one iodide from T4 and then you're going from T3 using the enzyme deiodinase. And these hormones will be stored in the colloid. And this is going to be sufficient for 2 to 3 months when it is produced. After coupling, the next thing will be thyroid hormone storage and secretion. After formation, thyroid hormones remain bound to thyroglobulin in the colloid until secreted. Can you see that T4 is still attached and T3 to the thyroglobulin? So hormones is surrounded in colloid by acid pool are then converted into colloid droplet. Thyroid stimulating hormone stimulates pinocytosis of the thyroglobulin into the follicular cell. When this hormone is released from the anterior pituitary to go and act on the thyroid gland, now it is going to lead to pinocytosis and the release of the hormone. Lysosomal enzymes will be able to hydrolyze peptide bonds and then release the T3 and T4 from the thyroglobulin. T3 and T4 will be discharged freely and secreted into the capillaries attaching to thyroxine binding globulin TBG. In patients with underactivity of the gland, increase in thyroxine binding globulin, there's going to be a decrease in free T3 and T4. Remember, these guys they have got underactivity of the gland, meaning that they don't produce much of T3 and T4. But then you have got a lot of the enzyme that bind to T3 and T4, meaning that the little T3 and T4 that you have will be bound to this enzyme. And what that means is that the body is going to interpret that you don't have any T3 and T4, so that is going to stimulate the release, okay, the release of the hormones from the anterior pituitary gland and above. So thyroid stimulating hormone will stimulate gland to secrete more of the hormone. So in other words, there is a feedback that is going back to the brain that you don't have enough of the gland. So the body is going to produce more of the thyroid stimulating hormone that will act more on the gland. And when you act more on the gland is going to start overworking, and that will make the gland to be big. That produces goiter. Okay, so that is how those are the steps. So this is the question as I told you that came. They said draw and describe the process of synthesis of thyroid hormone, 20 marks. You can see the steps here. Step 1, we have formed our thyroglobulin. Step 2, we trap the iodine. Step 3, we iodinize. Okay, there is iodination. There is iodination. And then step 4, we get the iodine, we add it to the T4, to the thyroglobulin. Step 5 will be now the coupling. We add the MITs and the MIT and DIT. Step 6 will be the storage. Step 7, we can release that into circulation. So step 7 will be deiodination of MIT and DIT. Okay, deiodination. So MIT and DIT are deiodinated inside the follicular cells by the enzyme thyroid deiodinase. Iodine, iodide, because it's an ion generated in this step, is going to be recycled so that it can be used again in the synthesis of more hormone. Tyrosine molecules are then going to be incorporated into the synthesis of new thyroglobulin to begin another cycle. So both the iodine and tyrosine are going to be salvaged. They are going to be used in another reaction by the enzyme so that you can form more of the hormone. A deficient of thyroid deiodinase or mimic dietary iodine deficiency. If you don't have the enzyme that breaks down the MIT and DIT, it will be more similar as though you don't have any home, you don't have any iodine because iodine cannot be released. So that is what happens. How do you control the secretion of the thyroid hormone? So the control of thyroid hormone. We have got the thyroid releasing hormone. We've already talked about this. Thyroid releasing hormone is released from the hypothalamus. To be specific, the hypothalamus, there is what is known as the paraventricular nucleus. That paraventricular nucleus of the hypothalamus is the one that is releasing the thyroid releasing hormone. When this hormone is released, it is going to go to the anterior pituitary gland and act on what we call the thyrotrophs. These are the cells, and this is going to cause the release of thyroid stimulating hormone, okay, from the anterior pituitary gland, from specifically the thyrotrophs. Thyroid stimulating hormone is a glycoprotein, and this guy is going to control the secretion of thyroid hormone. You can see what is happening. The pituitary gland, the paraventricular nucleus, is going to release thyroid releasing hormone, which acts on the thyrotrophs of the anterior pituitary gland to release the thyroid stimulating hormone, which causes the release of T3 and T4. These guys can now act in a negative feedback loop to inhibit this anterior pituitary gland. What are the functions of the thyroid stimulating hormone? To increase proteolysis of thyroglobulin, the breakdown of thyroglobulin, increase the pump activity for sodium and iodine. Remember, that was a function you needed to take in, the intake of the iodide. And then increase iodination of tyrosine, increase coupling. And then there's also trophic effects. Thyroid stimulating hormone secretion will actually start at 11 to 12 of gestational weeks. So in week 11 and week 12, that's when you start secreting the thyroid stimulating hormone. So this hormone is going to work via a GSC protein. We already know how a GS mechanism works up with. So the hormone will attach to the receptor. The receptor will activate the G protein. The G protein will activate adenylyl cyclase, which will convert ATP to CMP. CMP is now going to activate protein kinase A. Protein kinase A can phosphorylate protein, and that can lead now to the secretion of thyroid growth. It will lead to the thyroid growth. What are the factors that affect thyroid hormone secretion? You have got some factors that are stimulatory, others are inhibitory. On the stimulatory effects, thyroid stimulating hormone will stimulate the release of the hormone, and then thyroid stimulating immunoglobulins, these are antibodies. These are also going to stimulate the release of the hormone and then increase the thyroid binding globulin. For example, in pregnancy, this will increase. Remember, we said that this is going to attach to the thyroid, and the body interprets this that you don't have any hormone. So that is going to send a negative a feedback loop that is going to stimulate the production of more of the hormone. But inhibitory effects is if you don't have iodine, you will not produce. Okay, you will not produce. This is going to be an inhibitory effect. Actually, when you have got more iodine, it is an inhibitory factor. Okay, it is an inhibitory factor. But thyroid stimulating hormone is going to be released to cause the uptake of iodine. Okay, when you don't have the iodinase enzyme which produces more of the thyroid hormone, I mean more of the iodine by breaking down the thyroid hormone, the body is going to interpret that as deficiency of the iodine. And if you have got deficient of the iodine, the effect of that, you have got excess iodine intake. You start taking in more and more, and that's known as a Wolff-Chakoff effect. When you have got more of the thyroid stimulating immunoglobulins, which are similar to thyroid stimulating hormone, these ones can stimulate the gland to release more of the hormone, But the thyroid stimulating immunoglobulins, they don't give, and there is no negative feedback loop or on the secretion of the thyroid stimulating immunoglobulin. So meaning that these guys will continue stimulating the production of the thyroid hormone, and there's nothing to stop them. And that leads to hyperthyroidism, a lot of thyroid, and that's known as Graves' disease. Okay, so this is telling you the effect of thyroid hormone. Thyroid hormone is going to work on growth by stimulating growth formation, bone maturation, central nervous system for its maturation, body metabolic rate, basal metabolic rate by increasing the sodium potassium ATPase, oxygen consumption, heat production, and increasing the basal metabolic rate. Metabolism to increase the glucose absorption, the breakdown of glycogen, the making of glucose, the breakdown of the lipids, the synthesis of proteins. Basically, things that has to do with using more of your carbohydrates. On the cardiovascular system, to increase the cardiac output, more blood to go out of the heart. Okay, so this is just explaining the different functions that we have talked about. So clinical correlates, you have got hyperthyroidism. The most common hyperthyroidism is Graves' disease. Graves' disease is autoimmune, where there is a lot of antibodies, the thyroid stimulating antibodies. Now these guys resemble thyroid stimulating hormone. They also stimulate the production of thyroid hormone. But then there's no negative feedback on these guys. They'll continue producing the thyroid hormone more and more. So there is going to be hypertrophy of the gland because it is overworking. And other causes of hyperthyroidism are thyroid neoplasia. There is a cancer of the thyroid, and also overproduction of the thyroid releasing hormone from the hypothalamus and thyroid stimulating hormone from the pituitary gland. Okay, and also administration of excessive amount of exogenous thyroid hormone, like you are administering more of the thyroid hormone coming from the outside body. Hypothyroidism in adults is known as myxedema, and this is basically reduction of thyroid hormone. It can be primary or secondary. Primary just means that the disease is in the gland itself. For example, in Hashimoto's thyroiditis, you don't have iodine. That is because you don't have that enzyme, the deiodinase enzyme, that will be able to break down the thyroid to release the iodine. So what is the problem in, and this is how they brought last year. They brought a table and they put primary and secondary. They put T3, T4. You need just to put an arrow whether there is a decrease in T3 or there's an increase or there's no change in T3, T4, and the thyroid stimulating hormone. Just using an arrow, and it was a lot of marks. So in primary hypothyroidism, remember the problem is the gland itself, like in Hashimoto's disease. So because the problem is in the gland, there is a reduction in what the gland produces, that is T3 and T4. But because there's a reduction in T3 and T4 because the gland is not there to produce, there's going to be a feedback that is going to be sent to say, okay, anterior pituitary, can you produce? Maybe there's not enough thyroid stimulating hormone to produce the hormone. Can you produce more of it so that we can have more hormone? So the anterior pituitary will continue to produce thyroid stimulating hormone. So the thyroid stimulating hormone is going to increase. That will be reflex increase. But then the problem isn't even about this guy. The problem is about the gland itself. So this can help you to determine, if you measure the levels of T3 and T4 and also thyroid stimulating hormone, it will help you to determine where the problem is. So don't forget Hashimoto's disease. In secondary hypothyroidism, the disease is not in the gland, but it is starting in the higher centers, like in the pituitary or the thyroid. Sorry, the pituitary or the hypothalamus. So the hypothalamus itself has got lead to this decreased thyroid releasing hormone, and so there is a lead to production of the thyroid stimulating hormone, and that is also going to lead to T3 and T4. And so the follicular cells which produce T3 and T4 are going to be inactive. Okay. If you don't have any iodine, what happens? There's going to be a reduction in T3 and T4 because iodine is required in the production of these guys. But there's going to be increase in thyroid releasing hormone because that is going to send a negative feedback loop. And that is also going to lead to the increase in thyroid stimulating hormone. And that is going to lead to the increase in the gland because these hormones are going to stimulate the glands to be overworking. And that leads to what we call simple goiter. Simple goiter is because there was no iodine. And then if there is an absence of deionization enzyme, there's going to be no recycle synthesis of DIMT and MIT. And so these guys are going to be able to accumulate. DIMT and MIT will not be used for new thyroid hormone formation. And that is going to lead to the drop or the reduction in thyroid hormone because you can't use these guys because there's nothing to break them down so that you can reuse them. What are the symptoms if someone has got hypothyroidism? Just looking at the functions of the thyroid itself, there's going to be a decrease in basal metabolic rate. There's going to be a decrease in heart rate and pulse rate. There's going to be slow muscle contraction, reduction in appetite. Because remember, the thyroid gland is used in everything to do with excitement, where you use more energy. So there's going to be a reduction in appetite. But then there's going to be weight gain. Okay, because you are not using up a lot of carbohydrates in your body, you're not using them up, so meaning you're just storing them. That is why there's weight gain. There's also going to be constipation, prolonged sleep, dizziness, coarse skin, slow thinking, lethargy. Lethargy is where someone looks just weak, they can't do anything really. Mask face, intolerance to cold. Remember, the thyroid hormone is important in the generation of heat. So there's going to be intolerance to cold because there is a reduction in the ability to adapt to cold. And then there's also myxedema, swollen and puffy appearance of the body due to deposition of protein, carbohydrate complexes, mucopolysaccharide fluid in subcutaneous tissue. So someone is going to have the edema-like appearance, that's swelling. Hypothyroidism in children is known as creatinism. And in here, there's a reduction in T3 and T4 that will lead to reflex increase in thyroid stimulating hormone. Additional signs of hypothyroidism in children, there's going to be mental retardation, severe, short stature, because remember the thyroid hormone is also involved in growth. So to treat this condition, you can just give thyroxine, which is T4. Okay, so let's attempt some exam questions. What is the major product of the thyroid gland? T3, T4, calcitonin, iodine. Which one can you pick? Any other? So Josephine says D. So the major product is T4. How are thyroid hormones excreted mainly? In urine, sweat, stool, or saliva? So I'll include more questions. If you check the drive, I think there are questions there, more of them. Please look at them and go through them again and again. You can see that endocrine system is really easy. If you can just understand the functions of the hormone, If there is hyperactivity or hypoactivity, you'll be able to tell easily because you already know the function. So I'll end here. Next time we'll talk about the parathyroid gland.